Optimal health for high performers. This is the Health Upgrade Podcast with Dr. Nawaz Habib.
Hello, everybody. Welcome back to the Health Upgrade Podcast. This is Dr. Nawaz Habib, and I wanted to introduce this episode with a little bit of a disclaimer. We did have some minor technical difficulties while we were trying to get Dr. Rich Miller on the line with us. And so the beginning of the episode does begin with some minor technical difficulties. That said, we were honored to have Dr. Rich Miller join us. Dr. Miller is a professor of pathology at the University of Michigan and completed his MD and PhD at Yale in 1977. So he's very, very well versed in the areas of aging, and he's a major piece of the ITP puzzle. That's the Interventions Testing Program to determine which medications and which biochemical tools out there are wonderful for aging and longevity. During this episode, we talk about successes in lifespan extension, certain interventions that are currently under investigation, the importance of blood sugar levels, autonomic nervous system involvement, and the future of aging research. This was a really wonderful conversation between myself, JP, and Dr. Miller. Please excuse those initial interruptions from the technical difficulties, but as soon as we get started into that conversation, it's such a wonderful and enlightening conversation. I hope you guys take a lot away from it. Have a great day. All right, Dr. Miller, we're back. Let's continue that conversation so you can give us that overview of the ITP.
Well, the ITP stands for the Interventions Testing Program. We're just about to begin our 21st year, and NIA has agreed to continue to fund us for 5 more years. 3 sites participate, mine at Michigan, Randy Strong's at Texas San Antonio, and Ron Corstanja at the Jackson Lab, who's just replaced David Harrison, who was the bulwark of the program there for 20 years. The basic goal of the ITP is to see if there are drugs, and fortunately the answer turns out to be yes, which you can give to mice in their food, and it makes them live longer, healthier lives. Our first major success was the paper we published on rapamycin in 2009, although an earlier paper had shown good results for a different drug called NDGA. So the program was begun about 22 years ago. The NIA picked 3 labs that had good experience in doing mouse longevity and aging experiments. The program so far has found at least 4 drugs that extend lifespan by 15% or more. We have a recent paper with 2 others that seem to work, giving 8 to 12% increase. And we have a paper that's just been submitted with 2 others that worked very well, even started late in life. So the main idea here is both to see whether we can get ideas about drugs that might be useful in human preventive medicine, but I think at least as importantly at developing a repertoire of tools that we know can slow aging in mice and use those to see what are the common factors, the possible upstream pathways that are responsible for regulation, and can begin to use these to look at connections between aging and diseases like cancer and arthritis and dementia and all of the other things that make us more likely to be healthy at 25 than at 80.
So tell us about your model. You described using mice as the model, and has it always been the same model? And tell us a little bit about it.
Most people who do work with mice make the mistake of using a single inbred strain. The most typical one is C57 Black 6, an inbred strain in which not only are all the mice exactly identical, but they're all homozygous. That is, there's no genetic diversity even across the maternal and paternal chromosomes. So whatever they find using their Black 6 mice, they really have no idea whether it's going to apply to any other mouse in the universe. Sometimes it does, sometimes it doesn't. So we've never used that. We use a genetically heterogeneous mouse. Technically, the mouse is called PET3 because it was made at the University of Michigan and it's heterogeneous. Each mouse in our entire program, which is now up to about 28,000 mice, has the same genetic mother and the same genetic father, the same 2 parents. So just as you would share half of your genes at random with any brother or sister. Every mouse in our population shares half of its genes with every other mouse we've ever studied. So each mouse is genetically unique, but we can recreate the same population with the same average values and the same population structure at any time, at any size, in any laboratory in the world. That gives us the critical factor here, which is heterogeneity, but reproducible heterogeneity. This was a novel concept when introduced. It wasn't, our idea. It was actually developed by Tom Roderick and endorsed by the National Academy of Sciences in 1980, but everybody ignored those recommendations because they were committed to black 6 mice. Black 6 mice was sort of, as far as they knew, the mouse. And we've, I think, begun to convince people that that's a bad idea. And it's much smarter in almost all cases to use genetically heterogeneous animals. Now that we have lifespan and genetic and physiological and pathology data on, 28,000 mice, people are beginning to say, hey, wait, I shouldn't be using black 6 anymore. I should use those heterogeneous mice. The breakthrough last year was when Jackson Labs said, oh, yeah, maybe we should sell those. So that was good news.
So the genetic diversity in human beings is notoriously narrow compared with many other species. It sounds to me like the model you have has some level of genetic diversity that might approach what humans have. Is that a reasonable assumption?
Whether it's 50% or 75% or 125%, doesn't really matter. What really matters here is that can you draw robust inferences that are not dependent upon the accidental biology of one specific inbred mouse? So, there are tens of thousands of polymorphisms segregating within the Het3 population. Whether we would be smart to double that or triple it, it doesn't really make any difference. There's enough heterogeneity there that it's unlikely we're going to fool ourselves with false positive conclusions or false negative conclusions because of the lack of genetic diversity, which is true of sort of 95% of everybody else's mice.
So that explains how you chose the model. How do you choose the molecules that you're testing? This has been going on for a while, and what's the criteria, and has it changed or shifted over the years?
This is a community project, and it has been since the word go. Once a year, we publish an announcement saying, hey, you got any drugs you'd like us to test? Anybody who wants to, whether academic or commercial or a graduate student, can send in an application. It's a more or less free form. They tell us what drug they think we should test, 5 or 10 pages as to why they think we should test it. Sometimes that's cellular or molecular, or they have previous work in flies or mice or worms. They put forward their best case, and we ask them to tell us why they think it won't hurt the mice, because we want to do this with safety for the mice. Then we have a committee of 5 outside scientists, our access committee, read these and rank them. And then the final decision is made by the ITP's executive committee that consists of the 3 site directors, the NIA scientist, Dr. Fox, and also a committee of 5 outsiders who are not part of any of our labs. So we pick each year, we have enough money to test 5 or maybe 6 or occasionally 7 new compounds a year. So of the 20 or 25 applications that we get, we select a quarter, something like that, that convinces the members of our 2 different committees, this is where the action is most likely to lie. So we're making use of the brains and insights and intuitions of the entire research community. Now, they're very smart, very well-educated, experienced people, and so we have shown that the smart community of educated people gets it wrong about 85% of the time. It turns out to be, and I wouldn't do any better. I'd be happy if I got 15% of my guesses to be correct. It turns out to be kind of hard to guess what drugs will extend mouse lifespan. And you can say, look, 15% is very good. 20 years ago, the guess would have been, no, of course not. 0 compounds would do this. There's one of my favorite papers, which I give to all my graduate students, is a paper written by Linda Partridge and her colleagues in 1993, published in Nature, explaining with tons of math I don't understand why there could never, ever be a single gene or a single drug that would ever extend mouse lifespan. So a lot of people still think that might be true, and they're wrong. And so one of the actual sort of major global effects of the ITP is to slowly, gradually help people understand that if they think aging cannot be slowed, they've got that wrong. They need to put that aside and now think about projects in a universe in which aging in mammals can be slowed, and in a significant and dramatic way.
Well, and there's been work, I think most of them already know, about caloric restriction and how caloric.
That's right. That even in the 1990s, the caloric restriction papers were 60 years old, and when I argued with people saying, no, you can never have a drug that will slow aging, I would point to caloric restriction, saying you're sort of wrong, and we've known that since 1935, and that people were generally not able to put two and two together and see that argument. Well, there was an interesting episode at the Gordon Research Conference back in the mid-90s. George Martin, who's a terrific scientist, now deceased, a great friend of mine and of many of us, he asked the assembled experts, 150 of us, how likely they thought it would be that aging would be simple enough, controlled by an upstream process, that you could substantially slow aging in mammals by a drug or a single gene. This is just before Bartke famous publication in 1996 showing that it could be done. So 10 meant you were darn sure it was correct. That was my answer. 0 meant you were sure it could never, ever happen. That was George's answer. And these assembled experts, their average score was 3. So the notion back as early as the 90s, just before Bartke's paper, by people who certainly knew about caloric restriction, they had drawn the incorrect conclusion, almost all of them, that there was aging was too complex.
At this point, 12 hallmarks of aging, at least according to Lopez-Oten's paper from last year. Yes. They span everything from genetic instability and epigenetic drift to senescence and inflammaging and other things. The various molecules that you've tested over the now couple of decades of work span various different mechanisms. And so as we dive into the various different molecules that have and have not demonstrated that type of longevity enhancement, I'd love to get your take just to frame the conversation as to which of the hallmarks it affects. And of course, the hallmarks affect one another, so it's not always going to be quite so clean. But let's start with caloric restriction, which many people have looked at over nearly 100 years. There's the mechanistic target of rapamycin and nutrient sensing and whether or not you're triggering autophagy. There's been multiple different studies that have been conducted looking at it. What's your take on how caloric restriction actually functions to extend life?
Before I get to that, okay. I have 2 more global comments to make. One is to point out that I hate hallmarks. I think hallmarks are a major step backwards and a way of blunting thought. So I want to tell you why. There is no criterion for deciding whether your favorite assay or system or organelle or whatever deserves to be a hallmark. The principal way of this becoming a hallmark is to be something that the authors of that paper or the paper before that thought was kind of cool or kind of interesting, or they'd read a paper somewhere and they thought it might be fun to look at that. So being a hallmark, let's say you mitochondria or something, that's a hallmark. Who cares? It's not clear what that means. Does that mean mitochondria change with age? Or does it mean that by changing mitochondria, you can slow aging? Or have you been convinced by the kind of bad argument that you can accelerate aging by crushing mitochondria? Or did someone once publish an experiment in worms showing that by inhibiting some aspects of mitochondrial biology, the worms would live longer? So there's no plausible operational criterion other than somebody once had to write a review article and thought it might be fun to include this as a hallmark. So the criterion are awful. The other problem is, of course, that it closes thought. I think if you want to make an argument that senescent cells are important, which I think is a rotten argument, or the mitochondria is important, or the DNA repair is important, you should do that. You should look up the literature and make your best argument. But nowadays, unfortunately, people say, it's a hallmark. And so I don't have to justify it. I put in paragraph 1 of my grander paper, see, it's a hallmark. And that means that I and any sophisticated reader should accept that it's important. And to me, that's dodging your obligation to think about what way it's important, if it's important. The other reason is that the things that don't make it onto the hallmark list are officially branded losers. So if you're interested in another topic, membrane structure or something that the writers of your hallmark paper didn't happen to be thinking about the morning they sat down to make their list, you're in tough luck. And that's bad too, because it's really bad to sort of foreclose, even subconsciously. People, they don't see it as a hallmark, so they don't think about it. And that also is a mistake.
You stole my thunder. I was actually going to say that the reason why I was asking you to describe them in terms of hallmarks is to show that there are several of the molecules that don't work through any of the given 12 hallmarks.
So let me. I agree with you. So let me sort of make the second point, global point that was stimulated by your framing question there. One of the really great things about the ITP is that it allows us, for the first time, to ask the critical question, what are the common factors that these anti-aging interventions, including caloric restriction, and now a dozen or so others, have in common? So if one starts by saying, caloric restriction, I wonder how that works, you're stuck, because it does so many things, and there's no obvious criterion initially to sort of sort those out into those that are relevant to aging and those that are not. But what we can do, what my lab has spent the last 5 years doing basically, is starting to look for overlaps that are shared by mutants like the growth hormone receptor knockout and the Snell dwarf, by diets like methionine restriction and caloric restriction, and by drugs like the half a dozen potent ITP drugs. And the answer turns out to be there are some. So one of the ways we can start to think about your second half of your question, how does calorie restriction work to slow aging, is we can point out that, to pick one example, we have 13 of these, that UCP1 in mitochondria goes up, pro-inflammatory macrophages in fat go down, anti-inflammatory macrophages go up, CAP-independent translation goes up. There are several brain proteins involved in neurogenesis. They go up. They go up in caloric restriction. But more excitingly, they also go up in rapamycin mice, acarbose mice, mice treated with canagliflozin, mice with Snell dwarf or growth hormone receptor knockout. So once you've demonstrated for like a set of 10 different kinds of slow aging mice, that the same things happen, and they happen kind of quickly, by 4 to 6 months of age. It's not a matter of, the aging would make them go up and now they're going up slowly. It's instead a matter that the intervention changes them. And that also implies that they at least have some potential for being causative, the thing that actually is slowing the aging rate. So the great thing that the ITP provides us for the first time is lots of different ways of slowing aging, so that we can begin to ask answer cellular and molecular questions about which of the many things that calorie restriction does, or the Snell dwarf mutant does, or that rapamycin does, actually count.
Sounds like a sort of a machine learning approach, but with humans doing
it's not a machine learning mode. I beg to disagree with you. There are ways in which you can apply machine learning methods to this data set, and they're interesting to do. This is not this is an approach where one makes a guess as to something that might be important to aging, tests it in 10 different models of slowed aging, and you find out either you were right and it's changed in all 10 models, or you were wrong. The machine that did this is a postdoc.
Okay, let's talk about rapamycin, because that's one of the first of the molecules that really got heralded as a potential longevity enhancing opportunity for humans. It was discovered back in the 1950s or 60s in the soil samples of Easter Island. and it's actually an approved drug. It has so many different effects that it's been looked at as everything from an antibiotic to an anti-rejection drug. What did you see in the studies that you've conducted or that have been conducted by others?
Well, Dave Sharp at UT San Antonio made an application through our standard program saying you should test rapamycin, and his reasons for doing so were really good. In particular, people had shown that worms and fruit flies, if you knock out targeted rapamycin or if you inhibit it with drugs like rapamycin, they live longer. So he said it's also pretty potentially, at least, an anti-cancer drug. So Dave said, you should see if this extends mouse lifespan. And that was a very good guess, a very good intuition. So the initial paper, which came out in Nature, made two really interesting points. The first point it made was that you could extend mouse lifespan with rapamycin by 20%. 20% turns out to be a ton. To give you an example, if some magic pill cures cancer in people, and no one over the age of 50 ever gets cancer again, lifespan in humans will go up by 3%. And similarly, if you cure heart attacks and no human ever gets a heart attack, you also extend lifespan on average by 3%. This was published by J. Olshansky and Bruce Carnes and their colleagues in Science in 1990. So rapamycin by itself, at least in females, is giving 7 times better than you would get by a complete cure for cancer. And we've recently shown in a paper 2 years ago, if you combine two drugs, rapamycin plus a carbose, you can get in males a 29% extension. So that's 10-fold the amount of extension you would expect to see if there were no heart attacks or no cancers in people. So it's a big number. The other thing that was, I think, made our rapamycin paper important was that we found for the first time that giving a drug, even to middle-aged animals, had full potency. I found that astonishing, and I still don't understand it exactly, but that, I think, greatly augments the impact of the paper. My hunch was, I was confident of this wrong point, that in order for an anti-aging drug to work, you had to have it on board for most of your adult life, because you have to slow aging. You don't want to wait until aging really had caused major damage, and that by that time it might be too late. And that was wrong. Rapamycin, and it turns out all three of the other drugs of major impact, canagliflozin and 17-alpha-cetidiolinate carbose, if you start them in late life, they work also. A carbose gives 50% of the effect you would get if you start in young adults. The other two give 100% of the effect. That's important, not just theoretically, because it tells you there's some bad stuff going on, sensitive to anti-aging drugs, even when you're middle-aged, but it obviously has clinical implications. A lot of us are no longer in the first blush of youth, and a lot of the people who have congressional seats also are no longer very young, and so they have particular interest in agents that just might work at the median age of a congressperson.
I'm not sure we necessarily want to give them 30% longer to be in Congress, but
you'll have to be very careful. Yeah,
that suggests to me, and is consistent with the fact that many of the diseases of aging, things like cancer and heart disease that get worse, they accelerate in older age. You see the rise in the prevalence of cancer or incidence of cancer at different ages accelerates. It increases by about 1%
every 8 years.
So, but as you get, as you go from 70 to 80, the increase in that decade of risk is substantially more than the risk of cancer between 20 and 30. And so it seems to me that if you're working to reduce the risk of death at 60 or 70 or 80, by the same mechanism, you're just going to see a larger effect. But I want to make certain I understand the math here. What you're saying is, because sometimes this gets conflated in papers, if you were expecting the animal to live for a normalized 100 years, and you increase it by 29%, that means that the animal should then have an average life expectancy of 129. You're saying that it doesn't matter whether you start the treatment in year 30 or year 70 or year 80, either way, you're going to get to 129 so long as you've been using that therapy for some reasonable period of time before age 100. Is that correct?
So just for the record, we don't know if any of these work in people yet. Okay?
Fair enough.
You and I assume most people listening to the podcast fully understand that, but I want to make that point explicitly clear. So the average white American woman has a median survival of 80 years. So the best of our combination drugs that for females gives a 26% longevity increase. So if it worked in people, which we don't know, then the median survival of white American women would be about 100 years. And what that means is if you have a mental image of 40 different people age 80, some of them are really sick, some of them are still running in marathons, some of them are running labs, but some of them are retired and have some diseases. Well, that would be just the same spectrum, but shifted to 100 years of age, so that the average 80-year-old person might be similar to today's 65-year-old person, if these things work. Now, the mouse data suggests that 3 of our 4 drugs work if you give them to mice whose human equivalent age, that is in terms of the percentage of their lifespan they see behind them, is in their 50s or 60s. So it's not unreasonable to guess that if these drugs work in humans at all, you might start 50-year-old or 60-year-old people on these drugs and still see the same effect that you'd see if you'd started them when they were 30 or 40. That's a testable hypothesis.
Sure.
But we don't know if it's true or not. The conversation we were just having a moment ago about things that happen in your body soon after you get an anti-aging drug could potentially help address that issue. So that, for instance, right now, if you have a drug and you think it might slow aging in people, the obvious but impossible experiment is to take a batch of 40-year-olds, give them the drug, and come back in 40 years and see who's alive. That's the brute force way, and the obstacles to that, financial and temporal and logistical, are enormous. However, we now have good evidence that these drugs induce changes detectable in the blood, but also in the muscle and in the liver and in the fat of mice within at least 8 months of the time you start giving the drug. So if that's true in people, then you could take a drug that you thought might be an anti-aging drug in people. If it doesn't hurt them, give it to them for a year or two and see if these aging rate indicators have changed in ways that are consistent with the hypothesis that the drug is slowing the aging process. It's really tough to guess which drugs will work, but if you have a relatively short-term assay, an assay that takes, say, 6 months instead of 6 years or 60 years, to weed through the candidate drugs, the ideas to pick, if you find 2 or 3 where all of these aging rate indicators move to the favorable position, then those are the ones you really want to focus on in terms of trials of preventive medicines in people.
What are some of those age rate indicators that we're looking for in blood? And you mentioned liver and muscle as well. What are those markers that people generally could look at?
The fat starts to have more mitochondria and be bad at storing fat and good at making heat. Inflammation. Yeah, the white fat turns into brown fat in all 10 of the mouse models we've looked at. In addition, in all 10 models, the fat becomes less inflammatory. The inflammatory macrophages go down, the anti-inflammatory macrophages go up. In the muscle, there's a protein, it's called FNDC5, and what it does is it sends a signal to the fat that says turn brown. And the FNDC5 goes up in all 10 of these mouse models as well. Now, in the liver, there's a protein called GPLD1. There's a scientist in San Francisco, Saul Vieta, who's really put this on the map. What Saul's lab found was that if you exercise, either mouse or humans, if you exercise this protein goes up. The liver makes it; it goes up in the blood. So then furthermore found that if you make a mouse with lots of it, cognition is improved, which is a connection between exercise. That's why he did it, and exercise and good cognition. So what we did was we found that the GPLD1 goes up in the plasma and the liver and the fat, it turns out, for all 10 of our slow-aging mouse models, and the good things that happen in the brain, which involve a protein that protects brain cells from stress, called BDNF, and a protein in the brain that represents production of new neurons, that's called double cortin or DCX. Those go up in all 10 kinds of slow-aging mice, almost certainly because GPLD1 goes up. So that I've listed now brain stuff, muscle stuff, fat stuff, liver stuff, and plasma stuff, because the plasma molecule, GPLD1, can be detected in the plasma. One of our most important goals for the next year, if our grant gets funded, is to develop a longer list of things in the plasma that always go up in the slow-aging mice. We have made some strides in that direction. JP just mentioned machine learning. And in fact, there's a grad student in my lab who's using a machine learning method called the random forest method. What he has begun to do is look at plasma proteins and plasma metabolites from 7 kinds of slow-aging mice, not all 10, but we have data for 7 of them. And he has developed an algorithm based on the random forest approach that allows us to look at a tube of blood from a mouse given an unknown drug, and it'll tell you whether that plasma is like that of untreated mice or like that of mice that have been treated with an anti-aging drug. So we're now going to test his approach by doing the obvious thing. We're going to test some drugs that were not part of the data set he used to develop the algorithm, but drugs that we know either do or do not extend lifespan, because the ITP has just tested them. We haven't published the lifespan data yet. So if he's right, and if the approach worked, the green light will go on for all of the drugs that actually extended lifespan. They will produce metabolomic or proteomic changes consistent with this drug being an anti-aging drug. And the drugs that didn't work will resemble controlled plasma. That's our hope. We need to try that out in the next year or two, because that would greatly simplify both the translation of our ideas to humans, because humans have plasma, and you don't have to get their liver or brains out. You could just use plasma, and also give us new insights into the molecular pathways. That is, if a lot of the things that help the sorting algorithm, the random forest algorithm, discriminate winning drugs from loser drugs or controls, if a lot of them are turned out, and I'm going to make this up, this is complete fantasy, lipid metabolizing enzymes, or enzymes related to methionine metabolism or something like that, that really shines a flashlight on molecular cellular pathways we then need to investigate. Coming back to the main thread, you were asking what were, I've listed the cellular molecules, but there are also big molecular changes. 4 of them are worth mentioning. One is mTOR. mTOR is famous because of the rapamycin study, but it turns out all of these animals have lower mTOR in multiple tissues. That's Gonzalo Garcia's work. Gonzalo has also investigated 2 different MAP kinase pathways, one of which is enmeshed in inflammation, and the other of which controls protein translation. Both of these go up with aging, except in animals that have a slow aging mutation or have been treated with an anti-aging drug. So the TOR kinase, 2 different kinds of MAP kinase pathways. And the last is specialized translation. There are a handful of mRNAs that can be translated even when most translation is stopped. Most translation goes through a cap, a molecular structure at one end of the RNA. When you block that, which, for instance, rapamycin blocks that, a small handful of RNAs can be translated anyway by what's called cap-independent translation. And Gonzalo Garcia found that cap-independent translation is augmented by the genes that slow aging and by the drugs that slow aging. GPLD1, in a lovely piece of work, GPLD1 turns out to be a cap-independent translated protein. So that links Gonzalo's analyses of mRNA translation to Genet's analyses of the molecules that go up in the slow-aging mice. At least one of them, GPLD1, goes up because of selective mRNA translation. So others in the lab now are beginning to look at blood proteins that go up whenever you have a lot of cap-independent translation. That, too, may give us a bridge to human biology and human drug development.
Excellent. There's been a lot of talk about metformin. There's the TAME study that's being promoted. There's some controversy associated with the original work that was analyzed to suggest that metformin was providing diabetic patients with longevity enhancement. But there are other drugs for diabetes that have also been among the drugs that have shown longevity enhancement. You mentioned canagliflozin, I believe acarbose as well. Again, do you see, and maybe you've already answered this, but I'm just going to confirm, for example, the increase in beige or brown fat conversion, do you see that with those drugs as well?
So there are three good questions there. So let's do them one at a time. The original paper by Bannister on metformin, the epidemiology paper that alleged that diabetics on metformin had lower mortality risks than normal people, non-diabetic people, turns out to be wrong. Kåre Christensen and his study and his colleagues published a wonderful paper which has gotten a lot less publicity, both documenting that in the entire population of Denmark, including all the Danish twins, no such relationship exists, and pointing out the mistake in the arithmetic in the Bannister paper. So although that paper generated a great deal of enthusiasm and was one of the central pillars of the proponents of the TAME proposal, it's wrong.
Right.
And metformin does not actually extend lifespan in mice either. The ITP studied that in detail. Now, that doesn't mean that metformin might not be good for people, both diabetic and pre-diabetic and non-diabetic people. It might well be a terrific thing, but the intellectual support for making the argument that it will work in people as an anti-aging drug is a great deal weaker than originally proposed. If somebody gave me $50 million for a 5-year clinical trial, I would take the application for the TAME trial, remove the word metformin, and substitute canagliflozin, because the evidence for that, at least in the mouse studies, is a good deal stronger. And it's a drug that already has a great deal of clinical promise in not just diabetic, but non-diabetic people. So the second issue that you raised was...
Before you go there, it seems like that's an invitation to somebody out there to do the same work that was done in reanalyzing the Bannister paper with canagliflozin. I mean, there's got to be the opportunity to go to, maybe it's not Denmark this time, but go and do the work necessary to see whether or not what was being proposed as an effective metformin might actually exist in people on canagliflozin.
It's a complicated issue because usually canagliflozin is given to diabetics, and diabetics have a lot of comorbidities that are increased by the diabetes disease process. So, for instance, they have more cataracts. So a demonstration that canagliflozin retarded cataracts in a diabetic population would not be astonishing or novel. What you really want is canagliflozin given to normal volunteers. That's a research trial. You'd have to do that. Or even if you're going to look at epidemiological evidence, there are a lot of pre-diabetics who do not have exceptionally high blood sugar, but whose doctors were wise enough to give them canagliflozin in the hopes of retarding progression to frank diabetes. I don't know. I'm not a clinician or endocrinologist. I don't know how big that population is, but if I were into that, I would definitely want to look at pre-diabetics who'd been on canagliflozin for 5 years, who had not converted to diabetes. I would want to check how many of them got cancer, how many of them got Alzheimer's, how many of them got hearing loss or cataracts. I think that could be quite informative. Of course, we've done that in mice, but to do it in humans is now, I think, a high priority target. getting back to the canagliflozin acarbose part of your question, the data strikingly suggest that if you block the highest daily levels of glucose in mice, which is what both of these drugs do, they do it one in the kidney and one in the gut in terms of starch breakdown and glucose absorption, blocking not average daily glucose, but peak blood glucose seems to be extending your lifespan. And since most of the mice, about 80% of them, die of some kind of cancer, leukemia or breast cancer or liver cancer, whatever, some kind of cancer, the obvious implication is that keeping blood sugar quite low, not on average, but the peak blood sugar low, stops cancers. I've been trying to get my cancer biology colleagues to pay attention to this for about 20 years now. And I've kept nagging them by telling them we have anti-cancer drugs that prevent cancer. Rapamycin was the first, and now we have several that do it in a safe way in humans by preventing blood sugar rises that are too high. Wouldn't you like to have some anti-cancer drugs to study? Because you don't have any.
This sounds like Thomas Seyfried's arguments about glucose and Otto Warburg's observation about.
Leave that to one side for the moment, because sure. The key point here is that they don't care. Since the research came from an aging research community and not from the cancer biology community, they have no interest in that. It's a sort of a turf. If it came from a cancer biology lab, it's important. If it came from someone else, they want to take our money. And the same is true for my Alzheimer's friends and colleagues. If I suggest to them that by far the smartest way to minimize the damage done by Alzheimer's in human populations is to give them anti-aging drugs. They say, well, you're not an Alzheimer's person. You're trying to get Alzheimer's money. So I've had the late John Trojanowski. He was a terrific scientist, the director of aging and Alzheimer's research at Penn, argue with me in public saying, no, we want this money for Alzheimer's. Don't you think Alzheimer's is important? And I could not sort of make him see the light that, yeah, sure, it's important, but taking 5% of the Alzheimer's money and using it to test, to me, what is the obvious approach, which is slowing aging, and Alzheimer's will be slowed in parallel. He viewed that as an attempt to trick people into funding Alzheimer's disease at a lower level. It's a shame.
This is a very big shame, and it happens a lot in various different areas of medicine. If you bring something from outside that is A, cheap because it's a generic drug, and B, it works but was proven in something that really eviscerates their ability to get paid and to get funded, it gets rejected independent of its efficacy. That's disheartening.
Well, there are some exceptions. We've just, at Michigan, had the excellent fortune of recruiting a spectacular neuroscientist, Catherine Kazarowski. She's a good friend, and we're actually now starting to look at all sorts of cognitive changes in mice given ITP drugs, and it's about time.
Absolutely. Amen to that.
Based on everything that you've kind of just stated, I'm going to quickly summarize. It sounds like there's a metabolic basis to a lot of these chronic conditions. The metabolic basis of the amount of white fat to brown fat being linked to aging, being linked to diabetes, obviously, being a component of this, Alzheimer's, the metabolic basis of cancer as well, all having an important role to play here. Is that a fair summary assessment, that maintaining blood sugar levels without excessive levels of excessive spiking throughout the day, and caloric restriction being one of those tools that we can utilize, would be an effective method of maintaining
Let me both agree and
longevity
disagree with
or
you.
increasing longevity?
I definitely agree that there is a major thread throughout all of these conversations that modification of global metabolic pathways, including those that are linked to heat generation versus fat storage, and those that are linked to glucose, are usually, almost always, changed in these situations. A key exception, however, is rapamycin. Rapamycin, contrary to this sort of framework conception, leads to insulin resistance, eventually very poor glucose tolerance and very poor insulin tolerance. The other reason for disagreeing with this as the summary of what's going on here is that there are many other things that go wrong with aging, and that these drugs also block those. For instance, tendons. As you get older, your tendons get more cross-linked, so it's harder to stretch them, and if you do stretch them, they don't snap back. So we found that drugs, including rapamycin, retard both of those. The tendons of mice on rapamycin, Sue Brooks in Michigan found this. The tendons from these mice also are more resilient, younger, stretchier, more elastic. Cancer is dependent on a wide range of steps, mutations and evasions of immunity, et cetera, not all of which are safely called metabolic abnormalities or safely explained by the Warburg effect, which JP mentioned a moment ago. And cell death and production of inflammatory cytokines by cells which have been honored by being designated senescence, which is a whole different rant. Those are all changed by these anti-aging drugs, the anti-aging diets and anti-aging genes. So although modification of global as well as cellular metabolic pathways, I think is going to be a big part of the story, it's surely not the only part of the story. One can make an equally strong case for inflammation or control of cell proliferation or modification of neuroendocrine pathways or cell death or whatever. The fact that a lot of extracellular tissues like the lens of the eye and these tendons I just mentioned are also have their aging slowed down means that we're not sure yet what is the upstream thing that all of these drugs, all of these genetic mutations and all these diets are doing that then spreads out into changes in the fat, changes in the muscle, changes in MAP kinase, changes in TOR, changes in the brain. Figuring out what is upstream of these aging rate indicators is sort of the most important next milestone.
It seems like some of the drugs that have the effect of enhancing longevity, when combined, work to enhance even further the effectiveness. You mentioned rapamycin is about 20% when added with, I think it was canagliflozin. It goes up to about 29% in males, 26% in females. Yeah. So basically, with rapamycin combined with canagliflozin, you got an additive benefit. But I would imagine that, for example, acarbose and canagliflozin together don't. Is that what you're seeing, or have you not had the opportunity to combine them?
It's a really important question. So the trial of acarbose plus rapamycin was a rational one. Rapamycin, as I mentioned, impairs glucose tolerance. So one could make a theoretical case that acarbose would do a good thing even in rapamycin-treated mice because it tends to keep blood glucose low and doesn't challenge the beta cells in the pancreas quite so much. We suspected that it would do a good thing in males because the acarbose survival effect is really strong in males. It's significant in females, but it's smaller in females. So what we found in the males was that rapamycin plus acarbose gave you a 29% increase in median lifespan, whereas rapamycin by itself, at the dose we used, would have given only a 13%. So we went from 13% to 29%. And that 29% turns out to be the largest percentage increase we've ever seen for any drug or drug combination. Now, in the females, the small survival effect of acarbose could be because it doesn't work so well in females, or because it has a mixture of positive and negative effects in females. So if we had seen that rapamycin plus acarbose in the females did worse than rapamycin, we would blame that on toxic side effects of acarbose. If it did better, that would be terrific. As it happens, it did nothing. That is, rapamycin plus acarbose for females did the same as rapamycin by itself. So the implication is that using two drugs that are good for males does even better. So the more general question, what if you combine acarbose with 17-alpha-estradiol or rapamycin plus 17-alpha-estradiol? Those are good questions, and we are now indeed testing doubles of this general kind. Anyone who thinks they have a good rationale for combining two drugs is welcome to send us an application every February. And 2 of these have already gotten the seal of approval, and the trials are underway.
Well, let me ask about a couple of other drug classes where it seems that there's evidence to suggest that they have a longevity enhancing effect. One is in the senolytics area or senomorphics area. The combination of dasatinib and quercetin together seems to be providing that benefit. And the thesis is, or at least that's what is out there in the literature, that it's working by clearing senescent cells. What's your experience or the ITP experience with senolytics or senomorphics?
So I start from a position of great skepticism on senolytics in general and on the notion that there is such a thing as a senescent cell. I'm sure as you get older, you get lots of different kinds of cells with abnormalities in proliferation and or production of cytokines. That's cool, and it deserves investigation. To say, oh yeah, they are all the same thing. They are the famous senescent cell, strikes me as nuts. And the evidence for that is awful. And it's a terrible mistake because it closes off thinking. Now, as to the ITP's experience, Jim Kirkland, Paul Robbins, and several of their colleagues said, test fisetin. It's a great senolytic. Give it to the mice. Here's the dose to use. Here's the supplier to use. And here's 2 different regimens. One of these 2 is going to work. So we tried it. Neither regimen had any effect on mouse lifespan. Then we said to them, hey, wait, are you sure this removes senescent cells? Let's send you cells from our treated mice. We'll do them blind. You won't know which is control, which is treated. Test the number of senescent cells. And it turns out fisetin did not have any effect on senescent cells in the brain, in the liver, or in the muscle. So the dose that they believed was effective in mice as senolytic did not actually have any significant effect on senescent cells by any assay they chose to use. They used the beta-galactosidase assay, they used the P16 messenger RNA assay, they did a lot. And it's because fisetin didn't work.
Right.
Then the same group or a subset of them said, hey, well, okay, let's test dasatinib and quercetin. And since we'd been burned once, we said, okay, look, give us one published paper in which dasatinib and quercetin have been given in the food to normal mice and was shown to remove senescent cells. We don't want longevity data. That's asking for too much. All we want is proof that the drugs you want us to test, when given to mice in food, remove senescent cells. And it turns out there are no such papers. I wasn't able to find any, and more importantly, these experts were not able to find any. So there's a lot of commercial interest in the hope that there is such a thing as a senescent cell, that it's bad, that you could find a drug that removes, quote, the senescent cell. That will be good. But I don't basically trust papers from academic labs whose owners have a lot of money to be made, a lot of money to be gained by selling drugs for human consumption. Some of them may well be entirely honest. The data may be terrific, and I'm not going to believe it till it's reproduced by a lab that doesn't own a company.
Right, for sure. What about alpha-ketoglutarate?
Good question. So there's a famous paper, the one you're thinking of, in which they gave it to kind of some old black 6 mice, and it had a significant effect in one sex or another. So the ITP is testing it twice. Once we're recapitulating their protocol, starting with old mice. Of course, we would never use black 6 mice. Right. We are using het 3 mice because we don't care what it does to black 6 mice. And then in a second study, we're starting it in younger mice. The study with starting it in older mice is 3 deaths away from being complete. So we'll probably start running it up in a month once those last few mice die. The basic answer is that it didn't have a significant effect in males or females. We were not able to reproduce their effect in black 6 mice. Now, their effect was quite small. It was statistically significant, but it was in one sex only and was marginal in terms of the actual percentage increase. So it's not terribly surprising that a late start in our mice might not work. However, we're going to test it starting in earlier ages, and it's much too early to know if it's going to work or not. It's a good lead. We're trying it. We're following it up.
What was the thesis behind why, an intermediate in the citric acid cycle would be?
I don't know. I don't know the answer. You'd have to go to their paper and sort of see what the rationale was. My hunch, and it's only a hunch because I read the paper a couple of years ago, is that somebody found it worked well in worms, and said that there's an idea that if it works in worms, maybe it's worth testing in mice too.
Okay, so we've gone through quite a number of different drug classes. I think there's another one called meclizine or meclizine or something. That's
Meclizine, yes.
Meclizine. It's an mTOR inhibitor. Have you had any experience with it?
Sure. We just published a paper on it last year. It extends mouse lifespan by about 8% in males only. It did not have a significant effect on maximal longevity. Meclizine is available over the counter. That paper had 2 over-the-counter drugs, meclizine and astaxanthin, both of which extended male mouse lifespan to a significant effect. Meclizine is used to prevent seasickness. If you're on a long ocean voyage and you're prone to seasickness, there are over-the-counter preps, Bonine and several others, that help you avoid seasickness. It doesn't do that by an anti-aging effect, but it affects neurotransmitters. I don't know the detailed neurobiology of seasickness, but it certainly has potent CNS effects that blunt the seasickness, nausea and vomiting response. Now, Gino Cortopassi did a drug survey. He took some cells, dumped 6,000 drugs on them to ask which ones are the best at inhibiting mTOR. And meclizine was one of the 2 that emerged from that survey. So he recommended that the ITP tested, and it did have a significant, though so far quite small, effect on the male mouse survival. So we don't. If we want to check that out, we want to check it out at higher doses. Maybe it'll have a better effect at higher doses, or an effect on females at higher doses. If it does, we would then need to look carefully. Is it because it's an mTOR inhibitor? A reasonable guess. Or is it because of its well-characterized CNS effect, independent of whatever it does to mTOR, or a combination of both? That's a whole line of research that would be opened up once you become convinced that meclizine has an anti-aging effect.
Maybe tested in combination with rapamycin to see whether or not it has its effects are through a different mechanism. Because obviously, if it's through the mTOR, it might not have any additive effect.
Sure. Or test with a carbo.
Right. Many of these seem to have an effect on males, but not on females. Are there any that have an effect on females, but not males?
Not yet.
Interesting.
Rapamycin has approximately equal effect on both. It's a little tricky because the blood concentrations of rapamycin are higher in females than in males at any given chow dose. So if you normalize to blood concentrations, it's roughly equivalent in both sexes. Glycine, an amino acid, which we haven't mentioned in today's conversation, has an equivalent effect in males and in females. It's a really small effect. It's about a 5%, but it's there. It's seen in all 3 labs. It's seen in both sexes to an equal extent, and it's seen in rats. So it's probably real, but it is small and non-sex specific.
Are there any other amino acids that have been tested
Adding
for as well?
protein to the diet did not have a beneficial effect.
Okay.
Did it have a negative effect?
Interesting.
Have you seen any of the drugs that you've tested that have actually had a negative effect?
Yes, they're all in the last paper, the one that just came out a few months ago. And 2 of them are actually really interesting. You know already that canagliflozin has a positive effect in males, and when you start it early in life, it has no effect in females. Now, the new paper says if you start it late in life, it works great for males, and it has a negative effect in females, which took us by surprise. So it now seems plausible that canagliflozin has a mixture of positive and negative effects in females, and that the negative effects are seen in old age. So we're doing the obvious thing. We're going to start canagliflozin in females in youth, but stop it when they get to be middle-aged. The other reason why this idea is plausible is that if you look at any age at canagliflozin doses in the blood, they're 3 times higher in females than in males. And they also go up about 3-fold in old age, so that the blood levels of canagliflozin in the older females, that's where it's toxic, are like 9 to 20 times higher than in young males. So our hunch is, and this needs to be tested, it may not be correct, is that the negative effects of canagliflozin given to older females are because the levels are too high, and that maybe we can reveal latent positive effects by just not letting the females have it once they get too old. Has
anybody looked to see whether or not that is something that should be looked at in humans, for whether or not older females should tailor, scale back their dosing?
A question I plan to ask the next time I get in contact with a clinical diabetes expert. Yeah, it's a plausible bet. I'd be amazed if they hadn't done sex-specific surveys before they let it on the market, but I don't know the answer.
Yeah.
The other drug of special interest is 16-hydroxyestriol, not estradiol, estriol. We had found earlier that when you give 17-alpha estradiol, benefit for males, no effect for females. So we looked at the metabolites of steroids, and we found that there was a specific steroid, an estriol, that went up 20 times in male mice, but did not increase in females. In fact, in the drug-treated males, the estriols were far higher than in females, whether they had the drug or not. So this suggested the hypothesis, which turns out to be wrong, that the good stuff is the estriol, and that if you could give the estriols to the females, maybe the males can make it, the females can't make it. Maybe if you give it to the females, then suddenly you'll get a big positive effect. So we tested one of the estriols, 16-hydroxyestriol. When you give it to males, it's great. It's just as good, maybe even better than 17-alpha estradiol. If you give it to females, we thought it would work. In fact, it's toxic. It harms them. And we haven't a clue why that should be. It's not, in this case, due to higher blood levels. The blood levels are kind of similar in both males and females. So we don't know why this estriol is so good for males and so bad for females. The paper also had a third drug, a blood pressure medication, which doesn't do a thing for males, but is harmful slightly, but significantly harmful for females.
Interesting. One of the older techniques, which I guess was probably not the primary reason they did it, but castration of young males was something that they did for a variety of different reasons in hundreds of years ago, and there was some evidence, maybe apocryphal, that it had a benefit for longevity. Do you think that 17-alpha estradiol, I mean, this is a big stretch here, but giving 17-alpha estradiol to males has some effect that's in some way similar, that it's either anti-inflammatory or suppressing male hormones that are would otherwise be damaging, because females naturally live longer anyway. At least,
So let me say 2 things about that, or 3 things about it. The original idea was that if you give 17-alpha estradiol, which is a non-feminizing steroid,
right,
to males, they would live as long as females, for the same reason you were just implying. However, they live longer than females. So it's not a matter of recapitulating the lifespan advantage shown by normal females. It goes way beyond normal females. So it's not simply recapitulating the good news from estrogen. Secondly, we've given these drugs to castrated males, and we've given them to ovariectomized females. We didn't do a lifespan study. We studied instead some of the changes, like mTOR changes, and changes in exercise that the drugs induce. And it's really kind of interesting. If you give 17-alpha estradiol to males, does good things to mTOR and a couple of other things. This is Mike Garrett's work. But if the males have been castrated, you don't get that. Some of the effects of 17-alpha-estradiol in males on the biochemical indices of benefit are dependent upon a testicular hormone, presumably testosterone. The really cool thing came from the overrectomized females. If you give normal females 17-alpha-estradiol, we didn't see any effect. But if you gave it to overrectomized females, we did see an effect. So the implication here is that there are two things that are going on. In the males, the testosterone or something like it is needed. In the females, an ovarian hormone is actively suppressing the response to the 17-alpha-estradiol. So we would like to know how that works. Obviously, we would love to have drugs that work great in females. And one of our clues here is that the ovaries in normal females are producing something that makes the drug less efficient, at least 17-alpha-estradiol and acarbose, it turns out, less efficient in females than would be the case for overrectomized females.
So maybe, I mean, this also could be a big stretch. You would think to use it in women who are postmenopausal or have had a hysterectomy.
Instead of being a big stretch, that's a very good idea, yes. So these drugs might well work in older mice, for example, in which ovarian function is well below what it would be in youth. So yeah, it's a really good experiment.
Excellent. So how do we translate some of this from mice into other animal models and ultimately into humans? I know there's some work. I think there's a rapamycin study being done in companion animals, dogs. Are there other of these that are planned to be studied in animal models? And maybe before we even hit that, we have the challenge with many of these that there's very little pharmaceutical financial gain in all of these, because they're all either supplements or generic drugs or already marketed for other indications. And therefore, how are we going to, from an entrepreneurial standpoint, how are we going to get this out there and promoted once we do find one or more drugs that could be used in a cocktail or supplements? How are we going to do this? Is there a plan? Is there a?
So you've asked 3 or 4 questions, yeah. All of them good ones. Let's start at the beginning. The last 5 years, Matt Kaberlein and Dan Promislow and many of their colleagues have been studying dogs, dog aging, in the Dog Aging Project. A small subset of those dogs were being given rapamycin in a study that Matt Kaberlein was leading. This is the Dog Aging Project. Mm-hmm. I'm highly enthusiastic about the Dog Aging Project as a whole and about the intervention project itself as a subset of the Dog Aging Project. The reviewers of the grant that NIH just reviewed gave it a poor score, and the Aging Institute has declined to continue funding. So the Dog Aging Project as a whole is hanging by a thread, seeking private funding, trying to convince NIH to reconsider their decision, but it's not possible. It's I think not really possible to be terribly optimistic about that yet. The other thing where Matt and I disagree, I would not use rapamycin in dogs. I'd use canagliflozin because rapamycin, canagliflozin can be used in people for decades without hurting them, and it's not at all clear that people can take effective doses of rapamycin for a decade without harmful effects on their immune system, for example. So I think they're safer drugs that I would use if I were in charge of that chunk of the Dog Aging Project. Matt and I disagree about that. The other animal study that's underway is Adam Salmon's. He's at the UT San Antonio, and he's using common marmosets. It's a primate model, but it's a short-lived primate model with a median survival, I don't quite remember, 8, 9, 10, 11 years, something like that. So if rapamycin works in these marmosets, that will put to rest the at least arguably plausible concern that, it would work in mice, but not in a primate. So if Adam shows that it works in a primate, then that I think will remove one of the barriers to arguing it should be tested in people, or something should be tested in people. More importantly, in terms of science, before we get to the financial motivations and demotivators for this work, the scientific problem to solve in humans is obviously very, very tough. The lifespan studies, if you start in middle-aged, young or middle-aged people, are probably intractable. Even if you had an infinite supply of money, you don't have people wanting to sit around for 30 years waiting to see what will happen. So the companies that want to invest in this, they want to take a shortcut. They want to say, let's take some kind of old people and give them the drug and see what will happen in, 5 years, because I'm in a hurry. I'm going to retire and I want money for my stock options. So maybe it'll work in 5 years if you start with people who are old enough. And that's no longer silly, because some of these drugs do work in mice, in reasonably old, middle-aged or older mice, but it's making a guess that it would work in people who are already old. Some of them have died. Some of them already have cancer or some pre-diabetes or pre-senility or something. So that's one way of rushing things through. My answer to the problem, well, before we get to that, I think people will cheat. They'll go around the FDA. If we have, and we now have the first two, but we are likely to have more drugs that you can get at the local Kroger's or CVS or your local pharmacy, and the FDA has no control of that. That's going to get some of these drugs into people right away. In addition, if there is, let's say, firm evidence in monkeys or dogs and monkeys that rapamycin or canagliflozin increase your lifespan, healthy lifespan, by 10 or 20%, people are going to go to a country like India, which is not subject to the FDA's regulation, and buy it. Because the FDA will be viewed appropriately as an obstacle to health preservation under these circumstances. The last point I want to make is the one I made earlier, but because it's self-promotion, I like making it over and over again. The thing that's going to make a difference is having the ability to test potential anti-aging drugs in people without having to wait for 10 and 20 years of follow-up. If these aging rate indicators detectable in blood turn out to be kind of decent surrogates for the anti-aging state that is induced in mice by the different drugs and the diets and the single gene mutations, that for the first time will give you a broad open avenue towards human research. You can test a raft of drugs that are known to be safe for people or which are being prescribed for health conditions other than aging. And if it turns out that drugs A and B, but not C, D, E, and F, move the aging rate indicators to the favorable slow aging state, that's the first evidence you're going to have for saying, yeah, this might well work. This drug might well work in people. I think that'll make a big difference.
So all of the drugs that we've talked about today and all of the ones that I've seen published from the ITP, the goal is to enhance longevity. But in no case that I've been able to find are you looking at what I'm going to call sort of the other class of age-enhancing drugs, or therapies, which would be rejuvenating. And so I'm thinking now, and want to shift gears just briefly to the work that was originally started with Shinya Yamanaka and the OSKM factors of sort of reversing differentiation, sort of anti-differentiation, if you want to use the mathematical term, all the way back up to induced pluripotent stem cells. And the observation, I think it was Vittorio Sebastiano, who teased out that the shifting back of the epigenetic marks occurs sort of first in the process, or can be separable from the de-differentiation. Is ITP engaged in any aspect of work around what that mechanism is, whether it would be with small molecule drugs? I know that David Sinclair is racing around talking about the opportunity to potentially boil this down to one chemical that does all that versus, just something a little more basic, which is just using genetic triggers for OSK. Are you aware of that work being done? I'm sure you are. I would imagine you are. And are you guys doing any work in that area?
So let me say two things. The first one is easy. We don't know if these drugs, the ones we've already proven, do or do not rejuvenate. That is, the obvious experiments take some old mice, test them for something like muscle strength or cognition or visual acuity or hearing or whatever, or immunity. Give them the drugs for 4 months and then test them again and see if they have improved performance. It's not been done. That would answer the question of whether they are retarding aging per se or moving the mouse to a more useful state. So that's the first point. The second and more broader point has the rejuvenating factors. And it's an enormous amount of hype and salesmanship and faddish misunderstanding. The people who are selling this stuff, I mean, not just in terms of give me dollars and I'll give you a company, but trying to promote it in terms of science, they have a model of how aging works. Their model is when you're young, you have young cells. You get old, you have old cells. That's why you're in trouble, because all your cells are old. If I take all your old cells and I turn them into young cells, that will fix everything. This is ridiculous on its face, because, right, I mean, there are lots of cells that I had. There are cells that I had in my embryonic liver. I really needed them at the time because I needed to make a liver. But I don't want the cells in my current liver to go back to the embryonic state. A, they could become cancer. And B, the things they needed in the embryo to turn into really good, healthy baby liver, they're not there anymore. The neuroendocrine factors, the connective tissue matrices, the other cells to interact with. It's not a matter that cells start young, they become old. When you make them young again, that fixes everything. That's primary process thinking. That's the kind of thinking that a young child would have. It's been burned into the ethos of the aging research community by Leonard Hayflick and the people who bought the silly notion that the thing that happens to a diploid fibroblast in culture is aging. That the telomere-mediated loss of cellular proliferation, which is critically important in cancer, very obvious in human cultures, that's aging. Now, it has nothing to do with aging. If you look at some cells that can't divide, say, oh yeah, that's aging. It just doesn't add up. I'll tell you one quick story. This was told to me by Vittorio Defendi, who was at the time the chair of pathology at New York University. He was a colleague of Leonard Hayflick when Leonard was studying at the Worcester Institute trying to make viruses grow in human fibroblasts. Leonard would come down every couple of weeks griping, my cells were growing great last week, but this week they're just not growing. I don't know what, this is horrible. It's happened over and over again. And Vittorio said to him as a joke, maybe they're getting old, Len. Maybe they just got old. And Hayflick did not understand he was being joshed. He did not understand it was a joke. He thought it was an idea. And after a while, he decided it was his idea and that it was the correct idea. At that time, people were thrilled by cell culture. You could actually grow cells in culture. And the idea that you could study aging in culture without the mess of having mice and the time of waiting for them to get old, that was lovely to molecularly inclined cell biologists. They jumped on the silly idea that the cells were getting old in culture. And as you can easily see, this has transformed, I would say polluted and distracted an entire, now 2 generations of cellular biogerontologists. The current mode for that set of misordered thinking is that if I could get the cells to get young again, and I've got some factors I could sprinkle on, they'll get young again. That's going to fix stuff up. I was just at a meeting in which people presented some data. Nobody presented any data that showed that mice got healthier when you got some of their cells in the mouse body and made them younger. The most famous Sinclair paper, by sleight of hand, has convinced a lot of journalists that that's what he did. But of course, that's not what he did. He found ways of making mice older and did not actually test in public the notion that his rejuvenating factors would reverse or prevent that. The articles you see in popular science magazines and in USA Today show a picture of the old mouse, a picture of the young mouse. The old mouse looks terrible. The young mouse looks just terrific. And the reader, the casual reader, says, wow, he could turn that old mouse into the young mouse. But what Sinclair actually did, not in my view much of a triumph, is turn the young mouse into a very sick mouse that seems kind of old. He wasn't able, at least so far, to take a real old mouse and turn it into a young mouse, or to prevent aging from occurring. The last parts of the paper have to do with a genetic defect in a retinal cell, which these factors seem to be useful for, but that's not aging either.
So you're not a believer that reversing the epigenetic drift is an important stage of, or an important aspect of rejuvenating?
Well, that's a different question, entirely different question, and it's a good one. We don't know why aging causes liver cells and brain cells and blood cells and bone marrow cells and lots and lots and lots of other cells to start to do bad stuff, to make the wrong cytokines, or not to divide, or not to respond to the right signal, or whatever. We don't know how that happens. And although epigenetic drift is a sort of a broad cloud concept of many different things, some of them probably are involved in what goes wrong at the cellular level. Now, making that more precise, not just saying, see, it's epigenetic drift, but making it precise is an important therapeutic goal. If you find out, for instance, that the beta cells in your pancreas, as you get old, no longer have the ability to make a lot of insulin, you could ask, well, why is that? Are there changes in the signals coming into the cells, or the way they deal with the signals, or genes that they shouldn't be expressing? The same questions can be asked, probably with different answers, about the motor end plate that no longer provides adequate innervation of muscles, or the cells that are fat cells and invade muscles, or the cells in the bone marrow that no longer go to the thymus appropriately. All of those are questions for which hypotheses based upon disordered epigenetics are quite reasonable. But one needs to go beyond the sort of cloud of, yeah, it's all due to epigenetics, to, in this cell type, this specific change represents a change in this histone methylation, or this non-histone protein protecting you from methylation.
Well, I think we've just got a couple of minutes left, and I wanted to just bring it around to something that we do talk about a lot, which is how the autonomic nervous system impacts. And I know this is not your area of expertise, but a lot of the work that I've done, and that Dr. Habib has been involved in as well, is looking at how the autonomic nervous system affects inflammation and mitochondrial function. And so we do know, based on the work that's been done, largely spearheaded by Dr. Kevin Tracy, that there is a phenomenon called the immune reflex, and that by releasing acetylcholine and modulating T cell activity, it is possible to reduce inflammation levels, change the orientation of macrophages, and also to have an effect on mitochondrial function in a positive way, multiple different pathways. I realize you're not a big fan of the hallmarks of aging, and I understand why. And I'm also aware that there's a lot of overlap in the mechanisms by which those hallmarks do seem to have an effect on biology. Is that something that you think would be ripe for an application to the ITP?
Let me say two things. First, just because something is a hallmark of aging doesn't mean it's a bad idea. There are lots and lots of good things there,
Sure.
right? There's if you think that mitochondria are a hallmark of aging, or senescent cells are a hallmark of aging, or DNA damage is a hallmark of aging, it's very likely that studies of those are going to get you to important, interesting data sets. I'm just saying that you need to go beyond crowing that your guy became a hallmark and actually do something cool with it. Now, on the inflammation and autonomic nervous system, there are 2 points I want to make. I think the notion that the sympathetic nervous system or parasympathetic nervous system modulate age-associated changes in multiple tissues is a great idea. I don't know much about it. I was familiar maybe 20 years ago with the work done by David Felton, who was one of the first and strongest promoters of this broad notion. I thought David's work was terrific, and I was expecting to see others take it up and run with it, and I've lost track of how far that's gone. But the notion that age-associated changes in autonomic nervous function contribute to or lie behind lots of different kinds of cellular function, cellular dysfunction in old age, I think that's a great idea. And if this turns into ideas for drugs that might have a whole animal benefit, great. These would be excellent candidates for the ITP. The other notion that your comment triggered has to do with inflammation. There's a recent paper, came out of Nature last week from Stuart Cook's group, emphasizing interleukin 11 and its role in aging and aging biology. Cook's terrific, wonderful paper showed that interleukin 11 goes up with age in mice, and he took 3 different approaches to blunting it, one of them with an antibody and 2 of them involving genetic manipulations. And whenever you blocked interleukin 11, inflammation went down, which is expected, but more excitingly, lifespan went up. And all sorts of age-sensitive changes, for instance, changes in muscles, were also either retarded or reversed. It's a little hard to tell from the data in the paper. So the general notion that inflammation goes up with age and that's bad, that's only the starting point for thought. Papers like Cook and hopefully many successors that will take this hint start to move into specific molecular details. This cytokine in this cell type has this set of broad effects, and if I block that, I'll do good things. That goes well beyond the notion that inflammation is bad for you. Let's see what we can do with that. Similarly, questions about the autonomic nervous system are a good place to start thinking about, so what is the bad stuff they do, and can I block that specifically, and will that do good stuff? I think that's a really important question, but as far as I know, which is not very much, it's a question that's in need of a lot more data.
Yeah, so I think the question is it possible for the ITP to look beyond, and it is more work to do these types of studies, but look beyond a drug regimen to a device that stimulates the vagus nerve externally?
Nope.
It's not possible to do it. The work is just too heavy.
No, the ITP has a charter and a budget which gives drugs to mice, either in food or occasionally in water. We could consider applications that involve, say, one injection. But if one has an idea for an anti-aging intervention that is different, like a neurostimulator, just because the ITP can't do it doesn't mean to give up. One can, A, do it yourself, B, collaborate with a lab, maybe even an ITP lab that is interested in a collaboration. There are lots of things one can evaluate in terms of anti-aging interventions that do not make use of the ITP system and which could nonetheless be interesting, feasible, and potentially even fundable.
Okay, well, listen, I've got one last question as we sort of wrap this up, which is, what's your long-term vision for the ITP and the field of aging research in general? it's an open question. What would you like to see the field be doing 10, 15 years from now?
Well, the default answer, which I think is correct, but only partial, is I would like to see that in the next 10 or 15 years, the ITP will have documented in mice another dozen or so drugs that actually work, some of them in males and females, to slow aging. That's the essential goal of the ITP. But there are important things for aging researchers to do beyond that rubric. From my perspective, one of the most important potential consequences of the ITP is to rejigger how people think about preventive medicine. If you are wasting your time one disease at a time, you are focused on cancer or diabetes or cataracts. Instead, what you should be doing is working on aging, because it is by anti-aging manipulations that you're actually going to have an effect on the specific age-associated diseases that you've been spending your first half of your career on. So what I would like to see is an understanding in the medical research community and those who fund it generally that research on aging, its biology, and how to block or inhibit or even potentially reverse the effects of aging is far and away the most productive, most promising way to have a serious benefit to human health, much more so than one disease at a time research, however powerful and attractive and exciting and traditional that is. That would be the thing that will count. There's a whole intermediate level of ambition. The most obvious one is I would love to see a compact, plasma-based, well-validated set of aging research, aging rate indicators, things that change within a few months of the time that a real live anti-aging drug is given to a mouse. I'd like to prove that that also works in people, and I'd like to prove, all of this is speculation at this point, that this is a way of finding drugs that will slow aging in people. That's 15 years is sort of at the outer envelope for what you could do with that, but that's the line of ITP-inspired non-ITP research that I think has the most promise to bring the ITP's findings into as a central focus, into the main ring of human preventative medicine.
I think those are phenomenal goals, especially that second, third one. The idea of stopping the approach of masking symptoms and approaching one organ at a time or one disease state at a time, as opposed to looking at the really true underlying cause. And I think you're absolutely right that aging is.
I don't want to overstate it. If I have cancer, I want to talk to a doctor who knows an awful lot about cancer. And if I have diabetes or stroke, I want to talk to someone who knows. I'll read all those papers. But if I am more interested in how to prevent these things from happening in the first place, which I think is potentially a lot more productive, a lot more valuable. You don't prevent things by waiting for someone to get cancer and then cutting it out or killing the residual cells. To prevent cancer, you slow aging.
But your point earlier about glucose and making certain that you're recognizing the drugs that were used for other things and how they could be used, and I think that's something that's really important. So I just want to put an exclamation point.
Yeah, I agree.
But listen, I thank you so much for your time. I know that you weren't necessarily planning to spend a full 90 minutes with us, but I really appreciate that. It was an honor having you on. And Dr. Habib, any thoughts?
Yeah, this was an absolutely wonderful conversation. I learned a ton from both of you. This was great, but we really do want to thank you for your time, for your knowledge, and for sharing with our audience once again.
I appreciate the invitation. It's always fun to chat with people who know what they're talking about and who have good questions, who have thought their way through this. So please accept my thanks. And 2 years from now, if you want to repeat this, call me up. Let's do it.
Wonderful. We'd love to do that.
We will definitely do that. Thank you again. And for anybody who's still listening, share this episode with one person who you think could use this information, and get them to upgrade their health. Thank you so much for listening.